Welcome, everyone. My name is Daryl O'Sullivan with the Western Neuropathy Association, and I am very pleased to MC this new webinar where, as the Western Neuropathy Association attempts to provide ongoing webinar information and education to all the members and those caregivers who are interested in continuing to find information that will help those who are suffering from neuropathy and neuropathic pain. Today's presentation is a follow-up from previous presentations that related to Dr. Hammock and some of the work that he's done. I'd like to introduce and a great pleasure to introduce Dr. Karen Wagner. Dr. Wagner received her PhD at UC Davis in pharmacology and toxicology. Dr. Wagner is a researcher and colleague of Dr. Hammock. Her research focuses on neurobiology of pain as it relates to acute neuropathic pain and autonomic neuropathy. She is working to find effective medications that are non-opioid or narcotic, yet highly effective at reducing neuropathic pain. So today's presentation will provide us with an update on the EicOsis clinical trials. It is my great pleasure to introduce Karen Wagner. Dr. Wagner, it's all up to you now.
Okay, thanks so much. Thanks to Daryl, and I'd also like to thank Bev Anderson and the organizers for the invitation today to describe our research and clinical progress. As the title suggests, our technology targets the soluble epoxide hydrolase enzyme as a promising pain relief strategy. Oops, I'm having a little trouble advancing the slides. Let me just get over there. Okay, so I'd like to start today, I like to start my talks with some acknowledgements so that they're not lost in the shuffle at the end of presentations because they are very important. I'd like to thank Dr. Bruce Hammock, as Daryl mentioned. He's from the University of California, Davis, and he's also the CEO of EicOsis. He has led the drive tirelessly to translate this original science and research into a small molecule investigational new drug entity, which is currently in clinical trial. This also would not be possible without the help of open philanthropy and also NIH-sponsored funding programs, which include an important initial grant from the N9NDS Blueprint Program, as well as current HEAL grant from NIDA and NCI. That's the National Institute of Drug Abuse and also the National Cancer Institute. So with these funding agencies, we've been able to develop this as a new investigational drug and are currently processing through clinical trials with the FDA. I'll begin with a short overview of the arachidonic acid cascade and a history of the soluble epoxide hydrolase enzyme, which I'll refer to often as SEH. And then I'll outline our current approaches in targeting this enzyme as a novel way to combat neuropathic pain. The arachidonic acid itself is a lipid that is present in our cellular membranes and is released upon different types of stimulation. The arachidonic acid cascade is comprised of principally 3 enzymatic branches, including the cyclooxygenase, lipoxygenase, and cytochrome P450 pathways. The cyclooxygenase or COX pathway is perhaps the most well-known and the most well-exploited, as well as most NSAIDs target this pathway. That includes drugs like ibuprofen, Celebrex, aspirin even. The lipoxygenase or LOX enzymes are also well-known as they're, I might recognize them having leukotriene blockers. Those are the leukotrienes are the metabolite of LOX action. The CYP450 pathway has been more recently investigated and is the focus area of our research. The CYP450s, specifically monoepoxygenases of this family, metabolize parent polyunsaturated fatty acid substrates into epoxidized fatty acids. This is the example of an epoxy fatty acid. These epoxy fatty acids are the bioactive metabolites that are the subject of further metabolism and inactivation by the soluble epoxide hydrolase enzyme into what we call the dihydroxy byproducts, listed here as diheads. The soluble epoxide hydrolase enzyme was discovered roughly 40 years ago. Bruce Hammack was instrumental in that discovery. And after many years of research, it has been found to be a master regulator of these endogenous signaling lipids. It is a highly conserved enzyme, meaning it's found in species from plants to higher order mammals. And it also has a wide distribution in the body, but it actually presents in the highest concentrations in the liver, the kidney, followed by heart and lung. More importantly, for pain biology, SEH is also present in the central nervous system. It is in glia and neuronal cells, as well as in some astrocytes. The cascade is named after arachidonic acid, which is an omega-6 chain polyunsaturated fatty acid. However, it's been demonstrated also that omega-3 fatty acids such as DHA, docosahexaenoic acid, and EPA for all three of the same enzymatic pathways, and that includes the downstream SEH. The epoxidized metabolites of these parent PUFAs are polyunsaturated fatty acids, I'll abbreviate that to PUFAs, are bioactive, and in fact, DHA-derived metabolites may be the most potent, as they demonstrated the most potent analgesia in inflammatory pain models. The biological action of the EPFA as a group, so we're calling the epoxy fatty acids of all three classes EPFA, are largely beneficial. Their actions include analgesia, anti-inflammation, and in the case of EATS, well-known anti-hypertension. A common mechanism of the action of the EPFA and the SEH inhibitors among these biological responses is blocking endoplasmic reticulum stress. And for those of you who aren't familiar with that, endoplasmic reticulum is in the cell, and it's the place where most of the proteins are processed for export. And for cellular use. And when you have misfolded proteins, it causes a stress response. And so we limit that cellular stress response. Inhibiting the SEH has been the basis of several decades of research, which has led to tight binding, reversible transition state mimics that potently inhibit the SEH. And here, one of these in our target molecule for our IND candidate is EC5026. We are fortunate to have also several tools that include recombinant SEH enzymes of the mouse, rat, monkey, and human, as well as sensitive activity assays and mass spectrometry methods to analyze samples. That means for preclinical experiments, we are able to measure the in vivo exposure with sensitive mass spectrometry, looking at the exposure in animals to giving them the small molecule, and also the potency of these small molecules on the recombinant enzymes. So we can determine ahead of time how potent any new molecule is, and as well as when we dose it to an animal, look for it in the animal, and actually then correlate that to in vivo efficacy tests to know that we have exposure in the animal with the response that we're looking for. So the SEHI that we were able to use in vivo then demonstrated a robust response in inflammatory pain models in early investigations. Here, the SEH TPAU, so these are just acronyms for the type of molecule, and this is a different molecule, an earlier prototype than our IND candidate, but it exhibited dose-dependent analgesia in an LPS model in rats. So this is where we induce inflammation in the rat with LPS, which is endotoxin, if people are familiar with endotoxin, and then treat it with the inhibitors. We tested several of the inhibitors then in naive rats, naive rat controls, to see if this pain relief was actually related to any type of side effect like numbing or euphoria seen with other types of analgesics. In naive rat, we found that the inhibitors had no effect in the absence of pain using a von Frey assay. This result has been recapitulated in several models and several species over time, and therefore we are confident that with SEHI, we have no effect in the absence of a pain state. So this is different than a lot of current used therapies for analgesia, like opioids and others that give very, very active central nervous system side effects. We then further investigated the anti-inflammatory activity by testing different models of neuropathic pain, but the anti-inflammatory pain relievers have usually limited or no effect. Here we are seeing the chronic constriction injury in rat. So that is a ligature at the sciatic nerve. Very much gives a type of sciatica that people may be familiar with. And we saw a dose-dependent response of using EC5026 against this neuropathic pain. We then expanded these models to include using, I'm so sorry for that, to use pregabalin, which is the standard of care for neuropathy. And we saw that we had fold lower dosing giving exceedingly good response compared to pregabalin in this neuropathic pain model. At 1 hour, the EC5026 exceeded a 10 times higher dose of pregabalin, and it matched high dose. So that's pregabalin at 60 milligrams per kilogram compared to 5026 at 3 milligrams per kilogram in the chronic constriction injury neuropathy. Perhaps more notable was that the SEHI was active over the entire time course of the assay, which is pictured on the right, and it outlasted even the highest dose of pregabalin. Importantly, 5026 at this dose displayed none of the off-target effects on motor control that are known problems of high-dose gabapentinoids. For any of those in the crowd that take gabapentinoids, dizziness, sedation, there are all kinds of side effects that affect mobility and its use during the day. So to give a little bit of explanation with this slide, we test animals on a rotorod. So that's a rotating circular rod that they actually have to stay on and walk while it's rotating. And we found that, with a vehicle treatment, the animals can perform this really well. And with the 5026 and another candidate, 5029, which is our backup, at 3 mg per kg that was efficacious against the pain, we saw no changes or no statistical changes. But with gabapentin, the drug in question of the gabapentinoids, you saw them not being able to stay on the moving rotorod. Then on the right, you see that in terms of walking a balance beam, so this is where we actually have the rats walk down a balance beam where they are looked at for their latency to cross the beam or how many times they slipped their feet off of the beam, that the latency to cross and the foot slips were greatly increased by gabapentin. So the poor performance there, I forgive the arrow, that was for the other side, is actually in the increasing time to cross the beam. Then we moved to demonstrating efficacy and the unique characteristics of neuropathic pain with a few other types of assays. Demonstrating efficacy against these characteristics of neuropathic pain can be challenging in preclinical species as they can't speak to us. But we approach this by using operant assays where the behavior is operant, meaning voluntary, and it's reinforced by training. So these assays allow the test of conditions that are tonic, meaning they're ongoing, or even spontaneous and unrelated to stimulation that you saw in the former assays like Von Frey, where animals are actually probed with a filament. So this instead, the animals are not probed in any way, and they're just allowed to train with these environmental cues to assess more of the affective dimensions of pain. So we use the condition place preference assay to assess pain relief in the chronic diabetic neuropathy model. Briefly, the assay uses negative reinforcement, and the way we mean that is that we give the relief of the chronic pain as the reward, so to speak, to test the efficacy of the interventions. So the minus of being in pain is what actually is what they're seeking. And then rodents were trained for several days to associate the pain-relieving drug with environmental cues versus vehicle-paired alternative cues. The animals are tested then in a drug-free, they're tested drug-free, which is very important. So any of those types of secondary effects in the animal are not present. So they're tested drug-free in the open environment, and they're videoed without an investigator present, and then they're just assessed by video for their choice of cue, meaning where they spend their time in A or B in the open environment. And then the results are actually quantified as the change in time spent in the drug-paired environment, which indicates pain relief. So when we tested EC5026 in this paradigm, the increased time in the drug-paired chamber was indicated robust pain relief compared to vehicle. We've used this several times, and this is now used, like I said, in neuropathic mice. So this was the animals seeking pain relief with the administration of EC5026. In another paradigm, we actually used the same test and looked at the positive reinforcement in wild-type naive mice. So you'll see all the way on the left, in normal animals that are given the inhibitor, we checked whether or not they would have any, the small molecules would have any rewarding effects, whether or not the animals would seek it out in the absence of a pain state. And again, they did not. So then we went further, and then we made wild-type animals diabetic. So they had diabetic neuropathy. This is the middle panel. And when they had pain, then they sought out the pain relief. And we saw dose-dependent analgesia, which outperformed at the highest dose, outperformed gabapentin. And again, that performance is at a 10-fold lower dose than gabapentin. Then finally, we used yet again another paradigm, and this is even a little different again, where we used mice that have a knockout for the SEH enzyme, meaning they don't have the enzyme at all. And we gave the small molecule inhibitor, which targets that enzyme. And here we're looking to see whether or not whether or not there was any association between the small molecule and the target that maybe produced an off-target effect and was rewarding, and that's where it came from. But it showed here that there is no such association. So in animals lacking the enzyme, the inhibitor does absolutely nothing. But these mice that are genetically altered to lack the enzyme are completely sufficient to respond to morphine. And you can see that the positive control of morphine, which we know to be a very positive reinforcing drug that animals seek, gave a robust response. And that's on the last panel. So the SAHI had no effect in null mice, no effect in wild-type mice, but was potently analgesic in mice that had neuropathy. Additionally, analgesics such as the opioids used in the third panel induce drug seeking and tolerance in both naive and neuropathic animals. So, however, when we use the SEH inhibitors, they lack tolerance with repeated administration in chronic pain models, and they contrast greatly with opioids in that manner. Some of this work then led us to, as we explored how they are different from narcotics, also address what is a very urgent problem in the United States of the opioid withdrawal or opioid crisis, and looking at opioid withdrawal as something that we could treat with SEH inhibitors. So with the current opioid crisis, there's an urgent need for not only alternative analgesics, which 5026 is poised to meet, but also to treat the pain of opioid withdrawal, to help addicts or people who have used opioids chronically for pain conditions to actually wean off of opioids and maintain their abstinence as they experience withdrawal. For this experiment, rodents were made opioid dependent over 10 days of repeated administration. The opioids were then withheld, and they suffered acute cessation withdrawal pain, and that was normalized to 0. So at 0 on this graph, you'll see that animals are in the painful state. Then we dosed EC5026 orally, and the rats were tested over a short time course for their response. EC5026 effectively blocked the opioid withdrawal pain, and we find that this is promising to help addicts through their withdrawal side effects to stop using opioids. In another paradigm, another approach to limiting the number of opioid use disorders in the US and globally is to limit the need for opioids. We do this by dose sparing the opioids that are used, as well as providing an alternative to their use overall. So in this case, the SEH provide a unique opportunity for dose sparing because they don't overlap with the CNS depressant or the respiratory depressant side effects of other analgesics. Thus, where we show here on the left, we have morphine at low doses is actually at very low doses, they're subtherapeutic, they're not actually active in the neuropathic pain. And this again is using the chronic constriction injury model. But you'll also find that, you know, at times people will report that opioids aren't even effective at high dose for really relieving neuropathic pain. Here they were in the rodent, but you'll see on the right that as we give the same dose levels of morphine, that we can actually exceed the response with using a low dose EC5026 in combination with a low dose morphine. So we've been able to really limit the amount of morphine that the animals were exposed to and still get better than the high dose of morphine in terms of analgesic response out of the animals with this combination. And that's what we refer to as the dose sparing of opioids, where we can use a combination that actually exceeds the use of the drugs and gives very little opioid to the animals. So we now have these two approaches. And again, this is with the help of NIDA, the National Institute for Drug Abuse, and the HEAL program, funding to try and use EC5026 as an alternative analgesic to opioid use at all, but also to help people who have been exposed to opioids by limiting their withdrawal and actually limiting their actual dosing of those compounds. So just to give an overview, the mission of EicOsis is still to approach chronic pain as a single therapy, and we are making clinical trial progress towards this goal. The phase 1A clinical trial of EC5026 has completed, and that's the single ascending dose in healthy human volunteers, and it was dosed up to 24 milligrams in humans. I have included the clinicaltrial.gov identifier for anybody who would like to look at the details of that study. Just to outline what it was, is that it was a safety study in healthy volunteers, as mentioned, where oral EC5026 was given in 5 escalating doses to a total of 40 healthy male and female subjects. So in each cohort so the cohort is the dose level, 8 subjects of equal number, male and female, were dosed EC5026, 6 receiving the EC5026 and 2 being placebo controls. The dose escalation was done stepwise. So once we knew the safety and tolerability of the preceding dose, they would be, the next highest dose would be given. And the study subjects remained in the clinical research unit to have their PK and safety evaluated, and they were evaluated as outpatient through day 14. So we were able to actually monitor the compound in the blood, urine, and any kind of side effects over those 14 days with these healthy volunteers. And what I'm going to show next is kind of a complicated slide, but what it generally outlines is that we had good dose, we had good dose linearity, which is a highlight of any compound that the FDA likes to look at, meaning that as we increase the dose, we see the increased blood exposure in a linear fashion, and that we got good coverage for the effective dose of EC5026 with that, and then also a very as what our VP of Clinical Direction likes to say, that we had a very boring study, which is fantastic because there were very limited events that anybody noted, and none of them were drug related. You'll see too that we had good exposure over the mean time. So this is the area under the curve, which reflects how long the compound was in the body to cover that effective dose level. So we're getting very good exposure, dose linear exposure, and meeting what we think is going to be effective exposures for limiting pain in humans. So then that study is complete, and we have the data analyzed as demonstrated. But we have also now detailed our nested dual phase 1B trial approaches to the FDA. The dual phase 1b trial will test multiple ascending doses, first in healthy human volunteers, and then after a waiting period for the results on safety and review, the dose will be also tested in chronic pain patients to assess safety in a population with severe comorbidities and that are taking their standard of care medications. So we find this nested trial design really important because we would like to know how safe it is when we have patients that are on other therapies for their pain, and also whether or not they may or may not respond to the addition of the SEH inhibitor. So this might give us a very early readout on the positive effects, if any, on analgesia in these pain patients, as well as providing the safety data that the FDA wants to see. And we really do believe, this is some of the data that we've demonstrated before, and this is comes from a publication which is listed at the bottom. But we think there's a great potential for the SEHI to be effective in humans and bridge the gap from preclinical species to humans, because we have seen it be very effective in actual veterinary clinical conditions. So this is a horse that was being treated for laminitis. And laminitis is a chronic inflammation of the lamella sheath of the hoof, which ends up often being fatal because the animals cannot stand due to the pain. And then due to the hypotension of not standing, they have trouble eating and digesting, and it becomes a very chronic and fatal condition. But after the failure of standard of care drugs, as you can see on the left, the thoroughbred mare was about to be euthanized for the condition. When we had the approval to use an SEH inhibitor as compassionate use drug prior to euthanasia. She was given 5 days of treatment before we ran out of the drug. We were not prepared for treating a 1,500-pound animal, and it takes quite a lot of drug for a big pig basis. But after 5 days, actually, she recovered, she was able to eat, and she is alive and well today. So we found that to be bridging the gap of that species order in such a large mammal and orders different of preclinical species, and actually use in a real clinical presenting condition, although veterinary. It had all of the complications of a real presenting disease instead of a model, and we were able to help this animal. And so we really believe that that will translate to it being more broadly effective in humans as well. And then also, for one of our indications that we're proposing for the nested trial, not only for spinal pain, but also we're proposing to test a phase 1b nested trial in patients that have chronic osteoarthritis. And one of the reasons that we choose this is we have seen also in veterinary species, clinical veterinary species, that were quite effective in being able to treat osteoarthritis. So this is a picture of Polly, a horse that was suffering greatly with osteoarthritis. And I think it doesn't take clinical experience to notice how painful her gaiting is. And then later, oops, I'm sorry, I'm gonna have a little, let's try to advance. After treatment with the SEHI, so she was given treatment for 10 days. And this was at a very low dose, 0.1 mg per kg once a day of our SEH inhibitor, in addition to her standard of care, which was phenylbutazone. I'll just play that again for while I speak about it. So 10 days of treatment, and we had this kind of improvement in osteoarthritis in this large of a mammal with this much weight bearing. So we really believe that we may be able to translate this to human as well. And one of our phase 1b nested trial suggestions is to do osteoarthritis in humans. And that's also to use in humans that may be taking opioids for severe osteoarthritic pain. So for anyone who has further questions, I'd also like to direct you to our new website. We have a lot of the background information on our papers in the history of developing this technology throughout the years, and how we used it in not only neuropathic pain, but inflammatory pain. And then it also leads to some details of how it outperforms other drugs in terms of being good at antihypertension. So it's important to note when we talk about analgesics that NSAIDs, for one, are counterindicated for people who have hypertension because they affect the kidney, and they're not appropriate for long-time use. But that SEH inhibitors could offer an advantage there because not only are they potently analgesic, but they have advantages of having lowered hypertension in preclinical species as well. So with that, I know I've ended a little bit earlier, but hopefully we'll have lots of questions. And I'd like to thank everyone once again.
So we would like to ask you to go ahead and submit questions, and we'll do our best to coordinate them with Karen. In the meantime, Karen, would you like to actually show the cat video?
Certainly. And I think, forgive my technical difficulties, but I think before I actually skipped a slide or two, so I know we're done early, so we'll have a lot of time for this. So this was a cat that it was a clinical, preclinical model where we induced inflammation in the paw, which is quite visible. So that is the left hind paw is the paw that was affected by an inflammatory substance. And you can see the troubles in walking and climbing for the cat. And then we gave the compound, the same one we used in horse, to the cat and looked at the progression over time. And by 6 hours, the cat was recovered. I can just roll that again. So we're seeing in multiple orders of mammals that we're affecting inflammatory conditions in both neuropathic pain. So we think that because of the number of different species that respond and that the enzyme is in all of these mammals, that we'll have a very good potential to have it work in humans as well.
So I'll start with a question just in case. Again, maybe get some others to start thinking about some ideas. What's the likelihood of, or what's the future in terms of the clinical trial? And at what point will there be available for human consumption, so to speak?
That is, of course, entirely up to the FDA and how everything runs through. It is a time-consuming process. And I'll just, at the same time, maybe I'll pop up to our website so people can get more information. We're in early phases, so there's typically phase 1A and 1B are the safety trials. And when we complete those, then you'll go to phase 2, and those are actually trials of the efficacy in human patients. So we'd have to note that we run this through a contract research organization. So those are sometimes throughout the US, those sites. And so we don't actually have the opportunity to help people locally that are looking to enroll in pain studies. But those will be done at those research sites. And then once the phase 2 is completed, it would go into phase 3, and that's usually use in the chronic pain patients, and that's in the clinic under the authority and review of research of review boards. And so that's the kind of the outlay of how that progresses. So we're still in early stages, but we found it to be completely safe in the phase 1A. There were no adverse events. We had dose linear pharmacokinetics, as we mentioned. And we're hopeful that as we begin, we're slated to start the phase 1B in the end of this year. And that as that progresses, that we would follow up directly with the phase 2.
I now have a couple questions that I'd like to ask that are coming through. Do you envision the treatment to be continuing for life or one time only?
So we believe that this is an analgesic, meaning that you would take it routinely for your pain relief. The 1B studies that we're starting are called multiple ascending doses, which means that we would dose the people for 7 days to see that repeated dose administration and the safety of that. So this is, we don't, this is an analgesic. It's not necessarily a curative medication. To take it once and never have to take it again would be what would be a cure. But this is actually an analgesic. So yes, it is considered to be taken routinely for analgesia over the time needed.
Okay. Next question is, for the horse, it appears that the pain did not return. Was there regeneration of nerves or just super long-lasting pain relief?
So we'll say that the pain relief in the horse with laminitis, the pain relief was long enough that the inflammation in the lamina was able to resolve. So what happens with laminitis, it can happen at different severities where there's actual tissue destruction, kind of like if you tore a ligament, the ligament is always torn even though it feels better later because there's less, it's healed to some extent and there's no inflammation there, but the ligament is always a little bit weak if it was torn. That's kind of what happens if you consider that in the tissue lamina in the horse hoof. So although, and if you catch it early enough and you're able to treat it effectively enough, then there's not that actual structural damage, the horse can actually recover. So we think that basically we were able to help the horse recover enough that they were able to walk then on whatever remained of their intact structural integrity of their hoof and is fine.
And I think that answered the next question as well. Richard, if that's not the case, please let me know. Any other questions? So long term though, how long does it normally take when you have a drug trial? Could it take up to how many years to get this all complete?
Yeah, the drug trials take years. Years is not exceptional to say. And it really depends on the progress and any events. If it goes very quickly, there's no unexpected events that occur in the safety trials. Those would be completed, by early next year. And then when you go to efficacy trials, that depends on recruitment of patients. There have been challenges this year with clinical trials and the instance of COVID. In fact, we did have one patient that contracted COVID in the 14 days after our clinical trial and had to remove from the trial because he couldn't come back in for his blood draw. Those are unexpected details that can delay a trial, but we remain hopeful. And then it's to the review of all of the information to make sure there isn't anything unexpected and that we have potent analgesia on the order, but we're hopeful that we're only saying 2 years to seeing some of that efficacy.
Okay. All right. Now, who would actually, assuming that we got through the clinical trials and the FDA approved it, how would the drug actually be manufactured and then distributed?
So the drug is actually easy to manufacture. That's one of and again, it's one of these subtle, not very well described advantages of the SEH inhibitors. It's actually easy to manufacture, and it's actually been tableted for the clinical trials. So this would be, just a tablet formation that could be out from the pharmacy, basically in a bottle. Okay. And then those would be, we do have, I talked a little bit about the pharmacokinetics. I'm not sure about the, the technical side of things, how far to get into the science, but we do have a relatively long half-life. So we have, it's not on the order of you have to take it every 2 to 4 hours. We could probably have a once a day. We're hopeful for a once a day treatment. Okay.
I have another new question. Could you tell us about your special grant for cancer pain testing?
Absolutely. So recently we were looking on the research side of things to characterize the analgesia that we were finding with inhibiting SEH more broadly. And we did that in a number of different models. So we're looking at inflammatory pain, and neuropathic pain, and as I kind of described here, we've done things as different as a metabolic-based diabetic neuropathic pain, as well as a surgery-based that doesn't have the same metabolic factors of the chronic constriction injury. Another model that we've used is to induce neuropathy with chemotherapeutics. So people may be very familiar with cancer chemotherapy, especially Taxols and paclitaxel, or the taxanes and vincristines, some of these alkaloids that are used. A neuropathy develops based on their on-target effects on neurons. So any, quickly metabolizing cell is affected. And so the people actually get a neuropathy while they're getting their chemotherapy. And it sometimes can be dose limiting for the urgent need of their chemotherapy for their cancer. So we wanted to try and approach treating the pain of the neuropathy with, while people are on their chemotherapy so that they could actually continue treatment. And we've looked at several types of chemotherapy in rodent preclinical species, developed that pain with the same chemotherapeutic agents that are used in human, and treated the pain with SEH inhibitors, especially EC5026, and we found that it potently is analgesic against that type of pain. And so the grant that we have from NCI is to more fully characterize that and to actually look at what it would be to, if there are any issues with combining the SEH inhibitor with the chemotherapeutic for patients that have cancer.
Okay, I have another question. And this one says, do I understand correctly that this drug does not have any effects on the mu opioid receptor? This would be extremely important in helping patients who currently rely on opioid treatment.
That's true. So we've done a broad panel on what I'll call G protein coupled receptors, right? GPCR, if people know that, which include the opioid receptors, mu, kappa, and delta. We don't see any effects on those receptors. And what we know from using the SEH inhibitors extensively is that there aren't the similar type of CNS-related side effects. We don't see respiratory depression. That hasn't been quantified, but that's, from routine use in the laboratory in preclinical species, I would add. So we're not seeing the types of effects that you would see with mu opioid receptor agonism. And in fact, that happens to be what's complicated with some therapies is that the respiratory depression in combination with opioids, if it further accelerates that, it is problematic for patients, but we don't see that. So no, it does not, and we think that in combination it'll be a good alternative.
Good. Any other comments that you might want to talk about? Let's see, I got a raised hand here. I got to find out what that says. Okay. Not sure. I don't see it. So
just,
anyway,
oops,
I don't have any more current questions right now, unless you want anything to comment on.
No, just to review is that, we're looking forward to this December starting clinical trials, the phase 1B nested trials. We are targeting diabetic neuropathy and osteoarthritis as our first nested trials. That would mean the trials that go from healthy volunteers and then go into pain patients that are recruited for the clinical trial. After that, we're hopeful that we'll have some data that will substantiate its run in phase 2 trials, which are for efficacy, which would show the efficacious nature of the compounds in human. And then phase 3 is usually, like I said, in patients in the clinic with oversight. And that would be where we would, see where we're most effective.
Okay, now I have another question now. Then when do you think the trials like the one for the cancer pain will happen?
Well, we do have it proposed that that's a possible 1B nested trial. So we do work in collaboration with some centers that may be able to enroll cancer patients suffering neuropathy to try for this safety. And so that's not complete yet. That's still underway, but that if in terms that happens, that could be within the year.
Okay. Next question.
I'd say a year's duration. Let me say a year's duration. I know we're at the end of this year.
Another question, will the SEHI, the scihigh drug, also help reduce other symptoms of neuropathic pain, small fiber neuropathy, such as numbness, weakness, tingling, etc., hopefully reducing burning pain drastically?
I do believe that there's some evidence for reducing the burning pain. That's due to typically to the nociceptors. As far as tingling paresthesias, we don't have assays that we have investigated in that way. So we do believe that we limit, at the cellular level, that we limit ER stress, and we provide cellular support. So for the neuron itself, it's a possibility. I wouldn't say that we have direct evidence, but because we actually support cellular health, that would be possible. But I won't make the claim that we do that. But as far as burning pain and the mechanical and thermal allodynia, yes.
Good. Okay. So let's see. No, I'm still, I think so far we've covered all the questions. And how are we doing thinking for any other questions? So I'll give it a few more minutes and we'll see if we go on beyond that.
Sure.
I think this has been a very educational and very interesting update on what's happening, and also an overview of the whole process, and as well as the drug itself and all the different components of it, and how you've all done all the different testing. So I know a bit, and some of it's been really complicated, and some people have said it's been over their heads, but I think in the long run, the end of this, and the video should illustrate exactly what we're trying to get to. And the comments about the use of it instead of opioids is a significant factor for many people who suffer severe ongoing acute pain. So I think we're all just excited as can be, and we just wish it was out tomorrow, literally.
We've been, plugging ahead trying to make that happen. It is a long process, and we're very welcoming of the support from the Western Neuropathy Association. You have been very receptive early to our start as a company, and listening to our progress, and we appreciate that. It is a long road, and I know that we came out early with information about it as a possibility, and it's hard for people to wait out the times. It's hard for us as a company to spend the years it takes to actually get it on the to people as the potential with the potential that it has. But we really appreciate the support and people's interest along the way, and sticking with us.
Yeah, and of course, everyone wants to know when it'll be available for us, the patients. And as you actually have said very carefully, it's probably going to be a few years to as many as too many years. You just can't tell completely.
Right. To date, the positive news is that to date, we don't see a lot of adverse side effects, which would be the first things that would, slow progress. So we've completed the phase 1A. It was really just, nothing happened. That was remarkable, and that is actually fantastic. Like I said, we've started the 1B. It'd be great to see this in the 1B nested trials. So the 1B is healthy volunteers, and the nested is in pain patients. To see if that's safe with all of the other standard of care drugs that people have to take. That idea of assuring safety in combination is important. And so, but given the kind of profile that we have as an analgesic, that we don't have these overlapping problems with other medications, we think that actually, it's conjecture, but we feel that will help pace things along pretty nicely, that we won't see the unexpected problems that might crop up. And then, as that gets into the phase 2 studies where we see efficacy, then when we get that readout, that plugs ahead even further, and hopefully on a good pace.
Another question or comment is, what about fast track status given the prevalence?
I should add, we have recently applied, and we do have fast track status.
Oh, good. Excellent.
Right. The troubles have been, this is an unprecedented time in even federal agencies during COVID. So we have fast track status. We are in line with our clinical trials, and it's really just trying to keep things moving through systems that are a little bit hobbled by COVID and absences and things like that, and to keep going.
Mm-hmm. Okay, good. Okay. I have a call in user use raised hand, so we'll see what's happening here. Hopefully we'll get something here. Nope, not yet. Okay. Well, I think what we'll do is we'll sort of end the presentation. Wait a minute, I've got one more question here coming. Would you want to say anything about other things the lab is involved in? I know that Dr. Hammock has other projects going on.
Certainly. You know, this kind of is in line with the ER stress mechanism of action, and it's also in line with, in some ways, with the chemotherapy-induced neuropathy grant. But we have collaborations in the academic lab with Harvard, looking at a new idea that the cellular debris from surgery and chemotherapy actually can induce cytokine storm. So this is an idea that people may be familiar with if they've been reading the COVID literature, is the massive inflammation that can occur very rapidly, is what we could refer to as a cytokine storm. And there's new therapies like immune therapies like CAR-T in cancer that have this type of massive inflammatory event sometimes as a result of CAR-T therapy. And we think that, as you know, limiting ER stress and limiting inflammation, is that SCHI have been, at least in preclinical species, have demonstrated that they can limit that inflammation and provide a helpful support to both COVID in that manner, and limit and potentially limit the inflammatory events for people in ICU, as well as in cancer and CAR-T therapy, is limit that, you know, post-treatment massive upswing in inflammation. So we've actually, of course, you know, worked with some collaborations and have some grants in some of these areas as well. And this all stems from the anti-inflammatory nature of inhibiting SEH.
So another question is, will this video presentation be available to watch again on your website, the EicOsis.com website?
I believe it'll be available through your organization.
Yes, it will.
I don't know that, yes. So that's,
yeah.
But
okay.
We do have, you know, background papers and, you know, updates on our clinical trials and the kind of grants that we've had and the kind of research that we're doing on the website. As well as a portal to contact us with further questions.
Okay, I think that's pretty good.
Mm-hmm.
Okay, I think that I want to say thank you very much for the time and the excellent information and update. We, Western Neuropathy Association, our members and caregivers very much appreciate your time and effort and the dedication to helping those in acute pain and who really need the help. And the fact that we could do this without opioids would be phenomenal. So again, thank you very much for your time and effort, and wish you the best in terms of both your safety as well as your progress in making this happen.
And from all of us at EicOsis, we wish everyone, you know, health and safety in these times, as we mentioned, and, you know, that we are forever trying to make progress and get this to market so that we can provide people this type of analgesia. We do appreciate the struggles that patients face, you know, that sufferers of these chronic conditions face, and it just redoubles our efforts to get it through.
Yeah, and again, thank you. So just to let everyone know, the presentation will be on the Western Neuropathy Association website, and you can also use the EicOsis.com website to get more background information. And we'll be signing off now in terms of this particular webinar, but I want to remind you, next month we'll have another one. So look forward to hearing some information about what we're going to have in November. Again, thank you, and all of you have a very wonderful day. Goodbye. Thanks, Karen.
Thank you. Goodbye.
Bye-bye.